Our experiment was designed to test which conditions would allow a plant to be sustained. In order to test the plant's living conditions, there were three groups. The first group was the plot of plants with aphids present, and only aphids. The second plot had aphids and ladybugs, and the third plot was the control group, which had no added variables at all. Additionally, there was a fourth plant used to measure the dryness. Before the experiment was enacted, the plants all grew in the same environment, for the same amount of time and same climate conditions. The hypothesis was that if aphids, a parasite for plants, were introduced singularly to a plant, then the aphids would soon kill that plant because they were stealing all of the plant's nutrients. And with that hypothesis stated, soon came a second hypothesis saying that if we added a predator to plants with aphids, such as ladybugs, it would allow the nutrients stolen by the aphids to be decreased, and actually prolonging the life of the plant. Lastly, the control did not have any added variables to them, as stated before, and this allowed the experiment to provide a credible result about the different variables that are being tested in this experiment. Our main goal was to test how well the ladybugs worked when it came to promoting longevity in the plant's life. Now here's Kyla to talk about the methods that we used in our experiments.
Alright, so this is the method. For this experiment, the independent variable were the aphids and the lady beetles. And the dependent variables were the aphid population, so the number of aphids and plant growth. The controls were the soil, right here, type of plant, the seed, and time period of 8 weeks. We started by planting 2 of these beans, 2 of them, in 4 pots. We measured the growth in centimeters of each seed as it grew taller and bigger and bigger. We chose 3 of the pots and assigned them to either the control group, the aphid group, or the lady beetle group. We measured the plants weekly to see how they grew under these conditions. So for the first week, we measured it at this length. Second week at this length. Third week at this length, and so on. We began with 4 of these pots. So 4 of these each had 2 seeds each filled with the same type of soil, each with 2 little beans here. And they were left alone for about 2 weeks for growth, with watering and everything. So every plant was grown in the same environment and under the same conditions. Once growth was present, so once the beans have sprouted, little seeds there, we began measuring the growth rate of each of the 4 plants. So, see, one of the plants was smaller. Say we got a plant that was a little bit tinier, nothing on it. Say one had more leaves, throughout the whole process, and then one grew into a flower all the way to the top, whatever. In the 4th week, we then selected 3 out of the 4 pots and introduced 2 variables to these plants. The variables which would be ladybugs and the aphids. So one pot was selected to have aphids introduced. So we have one pot here, little aphids introduced to this plant. One pot also had ladybugs and aphids, so both of these variables in the plant there. And the 3rd plot was our control group, and it was just plant all by itself, which is ladybugs and aphids over the course of 8 weeks. So after the course of the 8 weeks, we will turn to the next video for the results.
So the graph shown in figure 1 is the correlation between plant height and plant dry mass based on 3-inch fava bean plants from the class experiment. The other plants were not included due to sampling error. As shown, there is a considerable variation in the data, making dry mass estimates unreliable for further use in the analysis. Figure 2 shows that average abundance of aphids counted per plant after the experiment. Error bars are standard error of the mean. Controlled plants contaminated with aphids were excluded from analysis. This analysis suggests a negative effect of lady beetles on aphid abundance. However, the general decline on both treatments suggests that the conditions were less than optimal at the end of the experiment. And finally, figure 3 shows the plant growth measured by height after experiment. The data suggests a negative effect of aphids on plant growth, both without and with lady beetles, in addition to the large variation in plant growth.
Although we were able to find that lady beetles had a negative effect on the growth of aphid population, there was also a decline from the group that only contained aphids. This alludes to the idea that the conditions this experiment were conducted under could be considered uninhabitable. This is a cause for possible experimental error. If this experiment were to be conducted again, extended research should be put into place about the conditions necessary for the survival of aphids. Likewise, by the end of the experiment, our group had reported that the 3 plants from each treatment died before we were supposed to conclude our measurements. The negative plant growth for these treatments, including negative growth in our control group, also gives a possibility of experimental error. By only conducting these measurements weekly, we left a large period of time that contained information that could have produced more conclusive results. In order to conduct this experiment to the best of its potential, we would need to measure each plant more frequently and make sure that there is sufficient watering and there is no other factors that would limit or create more error. By doing this, we would increase the chances of each plant's survival, as well as allowing ourselves a closer idea as to which plants survive the longest.